Hello, welcome back to Canna Intel 420. Today we're going somewhere most conversations about cannabis never actually start, the land itself. Not modern branding, not outcomes, not hype, but origin. Specifically, we're stepping back to Southern Siberia in the early 1920s, where a wild population of cannabis forced scientists to rethink how this plant adapts, survives, and evolves under pressure. Before we begin, a quick note for clarity. Everything discussed here is strictly educational. This is a botanical, historical, and ecological examination of a plant and its development over time. Nothing here is intended to promote or encourage use where it isn't legal, and nothing discussed should be taken as instruction. Laws vary by region, and respecting those boundaries matters. This series is about understanding cannabis as a biological system, how climate, geography, and survival pressures shape traits long before humans ever label them or commercialize them. If you've ever wondered why certain characteristics exist in the plant at all, this is where those answers actually begin. So with that context set, let's go back to where modern science first took notice. Southern Siberia, 1924. The ground underfoot is still cold from the night before, a thin crust of early spring frost clinging to the soil surface where the wind settles in sheets across the southern Siberian steppe. Even now, with the sun low and pale, the temperature barely presses above freezing. The horizon stretches without interruption, a wide continental plain shaped by glacial retreat, river migration, and the persistent abrasion of the climate. This is a landscape that does not soften easily. The winters arrive early, hold the land for long months, and release it only reluctantly. In 1924, Dmitrii Erastovich Janischevsky walked this terrain not as a wanderer, but as a trained observer, cataloging the botany of disturbed soils and agricultural margins in a region shaped by extreme seasonality. He would later write that the Volga-connected zones and the southern Siberian reaches offered one of the clearest living contrasts between cultivated hemp and the wild botanical forms that persisted where human attention, infrastructure, and abandonment intersected. Standing at the edge of a disused farm road, a narrow corridor where supply carts once passed, he could trace the geological logic of the place. The soil here was compacted but workable, a mix of loam, gravel, and wind-blown sediment. It was the kind of substrate that injures easily and repairs quickly, the exact conditions under which ruderal plants thrive. Broken ground invites colonizers, and the Siberian climate ensures only the hardiest survive long enough to leave seed. Janischevsky's training had prepared him to look for anomalies, the plants that do not behave exactly as their cultivated relatives do. What caught his attention that morning was not an impressive stand or an overwhelming presence. It was the opposite. The plants were small, almost forgettable at first glance, thin stems rising only a short distance before branching hesitantly, their leaves narrow and sparse, their form lacking the robust structure he associated with the industrial hemp varieties grown farther west. And yet, despite their modest size, they were not weak. Their stems had a tensile hardness unexpected for their diameter. Where most seedlings in this region lagged under the delayed warmth, these specimens held a steady vertical posture, as though they had already calibrated themselves to the short season ahead. He knelt to examine one more closely. The morphology diverged in subtle but consistent ways from the familiar cannabis sativa grown for fiber. The internodes were shorter. The branching pattern was minimal, conserving resources instead of spreading outward into multiple competing axes. The plant seemed architecturally designed for speed, every structure compressed, every decision oriented toward reaching reproductive readiness before the Siberian cold reclaimed the land. Janischevsky had seen hemp in more forgiving climates, tall, dense, and optimized for human cultivation. These wild plants, however, were optimized for survival in a region where the growing season was not a schedule but a narrow biological opportunity. The smell of the soil gave away its recent history. This was not untouched wilderness. It was disturbed ground, shaped by livestock movement, equipment tracks, and seasonal flooding. The botanist noted how these plants had taken advantage of the pattern. He observed the distribution, not in uniform rows, not in clumped stands typical of sown hemp, but scattered along the faint suggestions of former movement lines, evidence of a species adapting to the contours of human and environmental disruption alike. Ruderal species are defined by this behavior. They are the first to arrive when the land is damaged, and the last to leave when it stabilizes. These specimens matched that ecological profile with exacting clarity. He lifted a small echin, an individual seed, from the soil surface. It was different, smaller than typical cultivated hemp, with a particular curvature and surface patterning that stood out once he turned it toward the light. This was not simply a feral offshoot of agricultural plants left to struggle in the wild. It exhibited characteristics suggesting a population that had been self-sustaining for generations under harsh climatic pressures. The traits he recognized in the seed, stem, and foliage were too consistent to be chance remnants of cultivation. They reflected selection, natural, persistent, and shaped by the Siberian climate rather than by human intention. What he found that day would become the basis of a formal description, a cannabis population distinct enough in morphology, ecology, and development cycle to be recognized as its own botanical entity. This was not an agricultural variant, not a domesticated subspecies degraded by escape. It was something adapted far more specifically than that. Something that demonstrated a biological strategy he had not documented in cultivated hemp, an inherent acceleration of the life cycle that allowed the plant to evade the early freeze. In later observations, he would chart the behavior of these plants over time, noting how they advanced toward reproductive maturity based not on changes in daylight, but on an internal progression tied to age itself, a mechanism suited to environments where the sun's schedule cannot be trusted as a reliable cue. But in this moment, in the cold, thin light of the Southern Siberian morning, Janischevsky was not yet thinking in definitions. He was practicing the most fundamental act of science, recognizing that what stands before you does not match what you already know. The plant was familiar in outline, yet distinctly foreign in its details. It belonged to the genus he had studied extensively, but it obeyed constraints and opportunities that cultivated hemp never needed to confront. Here, beneath a sky that shortened summers into brief operational windows, a unique lineage had emerged, compact, fast, resilient, and unmistakably wild. The plants revealed their differences most clearly when Janischevsky shifted from broad observation to the measured, almost forensic attention that field taxonomy demands. He did not treat them as curiosities, but as specimens that required quantification: height, leaf geometry, branching pattern, echin morphology, stem composition, all recorded against the environmental data surrounding them. In the notebooks he carried, the emphasis was never on subjective impressions. It was on traits that could be described, compared, and replicated by any scientist who followed the same path into this part of southern Siberia. Standing in the disturbed margin of the field, he noted first the stature, short enough that the tallest plants barely reached his knee. This mattered because cultivated hemp of the era routinely exceeded 2 meters in height under good conditions. These wild individuals, by contrast, formed a population in which the majority held within a narrow vertical band. Their reduced height was not a symptom of malnutrition or crowding. The soil, though compacted, carried enough organic matter for taller growth if the genetics allowed it. The consistent small size pointed to a stable morphological pattern rather than an environmental deficiency. When he looked at the stem architecture, he saw a similarly consistent divergence. The stems were slender but firm, with a fibrous texture proportionally denser than that of cultivated hemp seedlings of equal diameter. Most individuals showed minimal lateral branching, often a single vertical stalk with only modest side growth near the nodes. This architecture matched a strategy he recognized from other ruderal species: reduce metabolic cost, prioritize upward development just enough to support reproductive structures, and avoid the excessive canopy expansion typical of domestic forms. Every structure was efficient, stripped of agricultural excess. The leaves confirmed the distinction further. Cannabis leaves are generically recognizable, palmate, serrated, but their proportions vary with lineage and adaptation. Here, the leaflets were narrow, with a surface area smaller than that of familiar fiber cultivars. The spacing between nodes was compressed, and the overall leaf arrangement lacked the broad photosynthetic surface preferred by plants bred for biomass. In a region where season length is brutally finite, these reduced leaves were not a flaw. They were an adaptation to maintain energy efficiency under prolonged periods of low temperatures and unpredictable light availability. Janischevsky recorded these observations with the same clinical neutrality he applied to any botanical subject, noting the morphology without attributing intention. At the soil line, he examined the root interface, carefully loosening a specimen to view the primary taproot. It was thicker than expected for a plant of its height, penetrating downward with a directness he recognized as a response to unstable surface layers. In disturbed soil ecosystems, shallow-rooted plants fail first. These individuals showed an anchoring strategy meant to resist displacement by wind, surface erosion, or the compaction forces left behind by agricultural machinery and livestock traffic. Again, not chaos, pattern. What held his attention longest, however, were the acons, the seeds that would eventually define the classification he proposed. Cultivated hemp aconies are often larger, with a surface pattern smooth or uniformly modeled depending on variety. These wild ones were smaller, with a distinctive modeling and curvature he documented meticulously. The coloration ranged from pale to dark brown, sometimes with a slight sheen. Many exhibited the drop-away trait he associated with natural dispersal timing. He recognized immediately that these were not the accidental byproducts of neglected crops. Their consistency implied a genetic lineage functioning independently of human cultivation pressures. He also took note of the distribution pattern across the site. The plants were not arranged in a manner that suggested deliberate sowing or agricultural escape from a single point. Instead, they followed micro-corridors, the edges of worn paths, the runoff line where seasonal meltwater pooled temporarily, and the low spots where carts or animals had overturned the soil. This distribution told him something essential. The plant's presence here was not static. It moved with disturbance. It exploited changes in the terrain. It behaved as a classic ruderal species does, colonizing damaged ground, surviving through rapid life cycles, and yielding seed that could remain viable until the next disruption opened the soil again. When he examined the developing inflorescences, he saw further confirmation of the distinction he was documenting. These early clusters were small, compact, and subdued compared to the resinous, highly selected structures of drug-type cannabis grown in more temperate climates. The reproductive structures here were functional above all else, built to mature quickly and persist just long enough to complete seed set before the temperature shifted. The plant's priorities were not aligned with agricultural or psychoactive optimization. They were aligned with survival under environmental extremity. He recorded the phenological cues as well. By this point in the year, cultivated hemp in milder regions of Russia would have displayed clear developmental benchmarks tied closely to the lengthening day. These plants, however, showed signs of progressing toward reproduction under conditions that would not normally trigger flowering in day-length sensitive cannabis. Though he could not yet prove it, the discrepancy implied an internal timing mechanism independent of photoperiod, an age-based progression that overrode the environmental norm. This observation, though preliminary, would become one of the defining scientific insights drawn from these populations. As he collected specimens for herbarium preparation, he also cataloged the environmental co-signatures surrounding each plant: soil temperature, moisture level, wind exposure, the presence of other pioneer species, and signs of past agricultural disturbance. He did not treat the plants as isolated organisms, but as components of a system shaped by cold stress, short summers, and recurring physical disruption. Only by understanding that context could he justify naming them as a distinct botanical entity. Nothing in what he observed suggested novelty for novelty's sake. The traits were not exaggerated. They were subtle, consistent, and biologically coherent. This was a plant performing exactly as natural selection would shape it in a region where biological timing determines survival. It was smaller because height provided no advantage here. It flowered quickly because delay meant extinction. It produced seeds built for resilience in unstable ground, and it persisted in places where human activity had been temporary, fragmented, or abandoned. Janischevsky did not yet call it cannabis ruderalis on this day, But he already understood before the formal description, before the classification debates that would follow, that he was looking at a population that had diverged not through human desire, but through environmental necessity. It was a botanical answer to the pressures of Southern Siberia, and the evidence was inscribed into every measurable structure of the plant. To understand why the plants Janischevsky documented differed so sharply from cultivated hemp, you have to step back from the individual specimen and look at the geography that shaped it. Southern Siberia, central Russia, and the western Siberian lowlands form one of the most demanding temperate biomes on Earth. This is not a region defined by steady seasons or predictable agricultural windows. It is a place where survival hinges on timing, where the temperature can shift rapidly, where the soil remains cold long into spring, and where the length of summer is not a guarantee but a narrow phase of biological opportunity. Seen from above, the region's structure is a layered map of influences. Central Russia's Volga Basin is a corridor of mixed forest and steppe, where human agriculture has existed for centuries but always contended with the climate's unpredictability. Western Siberia, extending eastward, is dominated by broad plains shaped by glacial movement and river systems that flood seasonally. The dominant theme connecting these territories is the short growing season. Even in years without extreme early freezes, plants may have only 100 to 120 frost-free days to germinate, grow, reproduce, and drop viable seed. Many years offer less. For a plant like cannabis, whose domesticated relatives typically rely on photoperiod to trigger reproduction, this environment creates a mismatch. The day-length cues that guide flowering in temperate latitudes do not align neatly with the truncated seasons of western Siberia. If a plant waits for the night to reach a specific length, it may never complete seed development before the cold returns. In regions farther west or south, the photoperiod system benefits the plant by aligning flowering with late summer when conditions favor reproductive success. But here, on the Siberian margins, that strategy becomes a liability. Timing is everything, and timing dictated by sunlight is unreliable. This is the selective pressure that shaped the populations Janischevsky observed. In these environments, the plants that survived were the ones that disconnected their reproductive cycle from photoperiod and instead relied on an internal developmental clock, a progression tied to age rather than to hours of darkness. This is not a mutation that appears suddenly across a population. It is the outcome of consistent environmental filtering. When early freezes kill individuals that flower too late, only those capable of initiating reproduction autonomously, without waiting for night length to guide them, manage to pass their genetics forward. Over generations, that advantage becomes fixed, a stable trait rather than a coincidence. When Janischevsky cataloged the range of these wild plants, he was not only documenting their physical location, he was documenting the boundaries of this evolutionary pressure. In the southwest, populations extended along the Volga's tributaries, where human disturbance created ideal conditions for ruderal colonization. Farther north and east, across the western Siberian steppe, the stands followed transportation lines, riverbanks, abandoned agricultural plots, and the fringes of forested zones. Every one of those environments shared the same defining constraint: summers too short for photoperiod-dependent cannabis to reliably complete a reproductive cycle. The climate data of these regions reinforces the logic. Winter temperatures routinely fall below -20 Celsius in many parts of western Siberia. Snow cover persists for 6 to 7 months of the year. Spring arrives late, often unpredictably, with nights returning to freezing temperatures long after the first thaw. Summer offers intense daylight, up to 17 or more hours at high latitudes, but the warmth does not last. Autumn descends abruptly, pulling soil temperatures back into ranges that halt root activity. Under these conditions, a plant that requires prolonged vegetative growth before flowering cannot secure its lineage. Natural selection rewards speed and penalizes hesitation. This is why the plants Janischevsky described matured quickly, remained compact, and initiated reproductive structures at a size that would seem premature in cultivated hemp. Their entire life cycle had shifted to align with the compressed seasonal window. Age-dependent flowering was not an incidental trait, it was an ecological necessity. The geography also contributed to the way these populations moved. Western Siberia's rivers, from the Irtysh to the Ob, create vast floodplains that are disturbed annually. Floodwater deposits silt, clears existing vegetation, and exposes bare ground ideal for ruderal colonizers. Wind-driven erosion and freeze-thaw cycles break apart the soil surface, generating microsites where seeds can establish. Human presence further accelerates disturbance. Transport routes, abandoned farming equipment, clearing of land, and livestock pressures create the exact substrate these plants require. This is why Ruderalis became so widespread across these territories. It was not merely surviving the environment, but exploiting every opportunity the landscape created. The same logic applies to the Volga Basin, where centuries of cultivation and transport have created continuous ribbons of disturbance. Wild cannabis in this region persists not by competing with established vegetation, but by filling the gaps in a constantly shifting mosaic of soils. When Janischevsky walked these borders, he was seeing the intersection of ecological pressure and evolutionary adaptation play out in real time. A plant that had broken from the photoperiod constraints of its relatives and established a strategy built around autonomy and urgency. Autoflowering is not an exotic trait. It is a direct and predictable response to the environmental rules of central Russia and western Siberia. Where the land shortens summer into a deadline, the plants that survive learn to ignore the signals of the sun and listen only to the progression of their own development. What appears today as a trait used in modern breeding began here as a quiet, functional adaptation in a region where waiting for nightfall could mean the difference between survival and extinction. This geographic origin, across the Volga corridor, the southern Siberian field zones, and the western reaches of the Siberian plain, is not simply where Ruderalis exists, it is where the trait that distinguishes it was forged. The map of its distribution is the map of its evolutionary pressure. And every plant Janischevsky collected in 1924 carried inside it the imprint of this terrain, this climate, and this relentless seasonal clock. By the time Janischevsky had observed these plants through multiple points in their development, one feature began to separate them decisively from the cultivated hemp he knew so well. It was not simply that they matured quickly. It was that their progression toward reproduction appeared indifferent to the external cues that governed most temperate plants of the genus. Where cultivated hemp delayed its transition until day length reached a specific threshold, these wild Siberian populations advanced on a schedule that seemed internal, almost indifferent to the changing length of daylight. To understand this distinction, it is necessary to set aside the language of breeding and return to first principles of plant physiology. Flowering is not a single event, but the result of a complex signaling network. In photoperiod-sensitive plants, this network is anchored to light perception, specifically the ratio of light to darkness sensed by photoreceptors in the leaves. This information is translated into hormonal signals that either permit or delay the transition from vegetative growth to reproductive development. In most domesticated forms of cannabis, this system is tightly coupled to seasonal light cycles, ensuring that flowering occurs when conditions historically favored seed maturation. In the environments Janischevsky was studying, that logic failed. Southern Siberia and the western Siberian plains do not provide reliable photoperiod alignment with temperature stability. Long daylight hours persist well into late summer, even as night temperatures drop rapidly and soil conditions deteriorate. A plant that waits for shorter days risks initiating reproduction too late to complete it. Under these conditions, the photoperiod pathway becomes maladaptive. Natural selection, indifferent to tradition, favors any genetic configuration that bypasses that delay. What Janischevsky was witnessing was the outcome of that pressure, a population in which flowering initiation was tied not to the length of the night, but to developmental age. The plant progressed through a fixed sequence: germination, early vegetative growth, structural establishment, and reproductive transition on a timeline governed internally rather than by the environment's light cues. This did not make the plant reckless, it made it predictable. In a landscape where external signals could not be trusted, internal timing became the safer strategy. The implications of this shift are profound when viewed through an ecological lens. Age-dependent flowering compresses the life cycle. Vegetative growth is minimized, not because biomass is unimportant, but because excess growth offers no advantage if the season will end abruptly. Resources are allocated early toward reproductive readiness. Internode spacing shortens. Branching is reduced. The plant becomes compact, not by accident, but by optimization. Each developmental stage follows the last without waiting for environmental permission. This mechanism also explains why these plants often initiate reproductive structures at a size that would appear premature in cultivated hemp. In agricultural contexts, size is associated with yield, and delayed flowering allows for greater accumulation of structural tissue. In Western Siberia, size is irrelevant if the plant does not complete its cycle. Survival is not measured in height, but in viable seed. Age-dependent flowering ensures that regardless of external fluctuations, reproduction begins early enough to succeed. From a physiological standpoint, this system likely involves altered regulation of flowering-associated genes, those responsible for integrating developmental signals rather than photoperiodic ones. While Janischevsky did not have access to molecular tools, his field observations align with what later genetic studies would suggest, that ruderal populations possess modifications in the pathways governing floral induction, allowing internal developmental cues to override external light signals. These changes did not arise to serve cultivation or human preference. They arose because the Siberian environment made them necessary. The climate reinforces this logic at every stage. Soil temperatures in early spring remain low, slowing initial growth. Sudden cold snaps can interrupt metabolic activity even after emergence. By late summer, the window for reproductive success begins to close rapidly as nights cool and frost risk returns. Under these conditions, any hesitation in developmental timing becomes a liability. Age-dependent flowering removes hesitation entirely. Once germination occurs, the clock starts, and the plant proceeds without pause. Janischevsky's records indicate that these plants did not behave erratically or unpredictably. Their timing was consistent within populations, suggesting that this was not a stress response but a heritable trait. Plants that relied on photoperiod cues would occasionally succeed in favorable years, but consistency favored the internally timed strategy. Over generations, the population stabilized around this mechanism, producing lineages for which flowering was no longer conditional but inevitable. It is important to understand that this life cycle strategy did not isolate the plant from its environment. Temperature, moisture, and soil conditions still influenced growth rate and overall vigor. What changed was the trigger for reproductive transition. The plant no longer asked the environment when it was time. It decided for itself. This distinction is what ultimately justified Janischevsky's proposal of a separate taxonomic identity. The plants he observed were not merely smaller or faster versions of cultivated hemp. They operated under a different developmental rule set, one shaped by the relentless arithmetic of a Siberian growing season. Age-dependent flowering was not a curiosity. It was the organizing principle of the plant's entire life history. By documenting this mechanism in situ, Janischevsky captured something that would not be fully appreciated until decades later, that the most consequential traits often arise not from abundance, but from constraint. In Western Siberia, constraint was absolute. The land allowed no margin for delay, and in response, these plants evolved a life cycle that ran on internal time alone. What we covered here isn't a strain story, and it isn't nostalgia. It's a plant doing exactly what biology demanded of it in one of the harshest growing environments on the planet. No mythology, no hype, just adaptation, pressure, and survival written into structure and timing. Russian Ruderalis didn't emerge because someone bred it that way. It emerged because Central Russia and Western Siberia left no room for hesitation. Short seasons, unreliable signals, and cold that doesn't negotiate. What you're looking at is a plant that learned to operate on its own clock because waiting meant failure. That's not a novelty trait. That's evolutionary logic, and that matters, especially now when so much conversation around cannabis skips straight to outcomes and forgets origins. If you don't understand where traits come from, geographically, climatically, biologically, you're only seeing the surface. This series is about going underneath that surface and staying there long enough for the picture to actually sharpen. As always, everything discussed here is presented for educational and informational purposes only. We're talking about botany, ecology, genetics, and history, not promotion, not instruction, and not encouragement of use where it isn't legal. Laws vary by state, country, and jurisdiction, and it's on all of us to respect that reality. To everyone who keeps showing up for this kind of content, the longer form, the slower pace, the deeper dives, I genuinely appreciate you. This channel exists because there are people who still care about understanding the plant instead of just labeling it. If that's you, you're in the right place. If you're new here, consider subscribing so you don't miss the next chapter. We'll keep building this out carefully, piece by piece, without shortcuts. And if you've been here for a while, thank you for sticking with work that takes its time and doesn't talk down to you. That's where we'll leave it for now. Until next time, stay curious, stay grounded, and take care of each other.